Yeah. Should we get started? Okay, so let's get started. I can't see Govind Swarup yet. So after Govind is Narasimhan. Narasimhan. Okay, so let me just. Oh, here's Govind. Indira. Yeah, it's okay. I'll get down in a second. Okay, my job is for just 30 seconds now to introduce Indira Chaudhary. Indira was introduced to us by Janaki Nair, who's somewhere here. Where did Janaki go? Yeah. And Indira's transformed from a professor of English to a historian of science, and she's done some really nice work, particularly on the history of TIFR and of Homi Bhabha, and she's done a huge amount of oral history on the protagonists of that time in TIFR, Obaid, PK Maitra, and several others. So she'll be moderating this session. So over to you, Indira. I'll be projecting on the screen. The people will be talking.
Thank you, Vijay. I think, as Vijay said, it was my privilege to interview a number of people who are going to be doing these interviews here, except for two. I think I've interviewed all of them. So I had the privilege of interviewing Obaid in the year between 2002 to 2003, and I did about 12 or 14 hours of interviews with him over 11 sessions, where I learned a lot, but learned especially about the beginnings of molecular biology, beginnings of his own scientific training. And I think this is a very interesting session to have. And we begin with Professor Govind Swarup telling us, who started around the time that Obaid started. And maybe he'll tell us a bit about, his own reminiscences of that time. Is there a microphone for him? Thank you. You have about 9 minutes. You can speak from below. Yeah, you can speak from there.
Speak next to you.
Sure.
Yeah, no, I'll speak very brief. Well, I'll be quite brief. Homi Bhabha, in his very important speech at Iksu in 1966, just before he died, January 6, I think, he made a statement how he built TIFR. He also said that if any young man with a proven competence were there, he hired them. One such person was Obaid Siddiqui, who joined 1962. 1962 early also, some of us had, from radio astronomers who were working across the world, had also written to him. He had also offered us. I joined in April 63. Now, what I consider extraordinary was that Kenilworth, whose picture was shown earlier today, there was 10-story building, and in which Homi Bhabha allotted 4 flats only to TIFR people. Maybe 5, I don't remember. Obaid, myself, Devendra Lal, and Daniel. 3 of us became FRS. So, what a coincidence. Is it Homi Bhabha, the influence that we felt very important, and therefore we had to work hard, his judgment. Then, of course, our children and Obaid's children were same age. We were in their house all the time. We have a great nostalgic memory, Bina and myself, about Obaid, those years. And later on, I had many other interactions with Obaid, but I would like to tell you another interaction was that in 1991 or so, 1990 or 1991, I proposed formation of National Center Radio Astrophysics, Autonomous Center under TIFR. Okay. And Obaid also, when I talked to him, he also thought, yes, it's a good idea. We had already started a center at Pune, Obaid was thinking also to become autonomous and move away from Bombay. There was not enough space. Then their director, TFR, was hesitant about the concept. So I went to Delhi, where Professor Menon was there. I talked to him. He said, yes. So he was strongly supportive. He asked us to attend, I asked me to come from Pune to Bombay when the next council meeting was there. There's a long story, I will not quite fully tell you that, but he invited us to there. Obaid was hesitant for half an hour. I had to discuss with him. He said, well, but Virender Singh had not invited him, so how can we go to the council meeting? Do you recall that? I said, well, Menon has said there. So we went there, and we, of course, J.R.D. Tata said, why one national center? TFR should have many national centers. That's what brought the beginning of NCBS. Today it's a great institution, and thanks to nurturing of Obaid, and of course now carrying torch by Vijayaraghavan. So I'm most delighted to be here. I have a lot of nostalgic memory. I should also add there that we had Devendra Lal, myself and Obaid, while we were in Kenilworth building, we had a carpool. So I had a lot of discussions, lot of memories. What he said between physics and biology, what we do. He said, what he was telling me today also, in astronomy you're tied up with big instruments. But in biology you can plan, experiment every new day, every new experiment. It's a great science. I think if I was a young man today, K.S. Krishnan, the Raman Krishnan Nobel Prize paper, taught me BSc first year electromagnetism. I worked in parametric resonance for 3 years, and I asked him, will it make a discovery? He sent me to Australia where radio astronomy was being discovered. Today, if I was a young man, I'll join biology. Thank you so much.
Thank you, Professor Swarup. I think one of the things I will share with you is that when I was interviewing Professor R.R. Daniel, who is no more, he spoke about how most physicists could relate to radio astronomy, but they didn't think that really biology should become a part of TIFR. And then they were glad when they saw it come up. But the other field, which was there very much in TIFR and very strongly so, was mathematics. And may I now invite Professor Narasimhan to share his memories of Obaid and biology.
I've known Obaid for more than 50 years. And actually, as Govind Swarup was saying, I got to know him better during the trip from TIFR to the Bombay airport and back. We used to share the same car, from the TIFR campus to airport or vice versa. And that's where we used to have very long and intense conversations. And probably that's where I got to know him much better than other interactions, of course, I had in TIFR. And also by talking to him, I could see how cultured he was, not only in his subject, one could talk about different things, about politics, history, and of course, culture. Now, mathematicians are reputed to be very absent-minded. And the classic example of absent-minded professor is a mathematician. But I was very happy to see a mathematician who was also absent-minded, at least who appeared to be absent-minded. Now, I can tell you, sorry. Well, sure, I'm a mathematician, right? Absolutely. Okay. So I'll give you some examples. Once we turned up at the Bombay airport, morning at, 5 or 6 o'clock or something, and we go to the check-in counter, and Obaid presents a ticket. Then the person says, but this is not the ticket for this flight. This is for an earlier flight which is already your travel. And, it was not, nowadays you can go and print it out couldn't do that. There were nice tickets and so on. But I must say that using his charm, he persuaded the people in the counter to let him board the flight. This is absolutely true. And again, once I remember we almost missed a flight. We were waiting to board or something, and he was so intensely talking about something that you wouldn't even pay any attention to the announcements. And I was all the time very nervous and finally almost to drag him, let's go into the plane. Now, more seriously, probably what absent-mindedness or whatever means that a person can concentrate at least for a certain extended period on something, some problem or some other question, forgetting your environment. This is probably how you achieve in your science and also in institutional building. You have to think about things, and at the time you have to forget, well, maybe your family and your environment. That's a kind of positive aspect of being absent-minded. So after he moved to Bangalore, I didn't have much contact with him. But however, I used to meet him occasionally. Once I remember he came to Trieste to organize a workshop or a school as a director. And we had long conversations how to maximize the use of ICTP for scientists, especially for biologists from the third world. And as again Swarup was saying, our children grew up at the same time. So our families have been very close, and especially the daughters grew up together and still maintain very close contact, which are part of the world they live. And that's a very important sort of component in our lives. And finally, I would like to wish Homi all the best in the years to come.
Well, moving from one mathematician to another, may I now request Professor Raghunathan to say a few words.
I was happy to be here on this occasion. I have known Obaid for a long time too. When Obaid joined the faculty at the Chaudhary Institute, I was a student of mathematics, of course. And at that time, I can't say I had much contact with him to begin with. In fact, I held him in great awe because of the reputation he had as a scientist. And we were all told that Bhabha had specifically to build a biology group. And that was, of course, something which made us, it's very impressive. But there's something else which impressed me very greatly. It was that I was told that he had an encounter with Jawaharlal Nehru as a rebellious student. That impressed me greatly. Anyway, so, but it's in the 70s really that I got to know him much better. And my first contacts were probably on the tennis courts. He was, of course, a superb tennis player and with a very elegant style. And I used to watch him. It was a great pleasure watching him. And occasionally I got a chance to play with him, which made me very happy. I'm not sure that he found the encounters equally a happy circumstance. I was a poor player, to put it mildly. So he couldn't have enjoyed playing with me, but I enjoyed playing with him. Well, our contacts elsewhere were mostly social. I'm a mathematician and very far from the kind of science he does. So we really didn't have any contact by way of science. But we used to talk about other things. I always enjoyed joining him in a conversation, either at the AFR canteen or on walks along the seashore. This was a slightly more equal encounter, unlike the tennis courts. And because I heard lots of interesting things from him about various subjects, science in particular, about science and scientists. Here, as I said, I was on more equal terms because I had a reasonable repertoire of gossip about science and scientists. So, well, as I said, it was always very enjoyable. But the contacts were really not not at a professional level at all. I mean, because the mathematicians had their own faculty and they met together, and the physicists, which of course encompassed biology as well, had a separate meetings of their faculty. So it was not, there was no such interaction. There is one occasion, however, when there was some kind of interaction in matters relating to the institute. And Professor Menon left the Tata Institute, Obaid, along with R. Narasimhan, took the initiative to organize a group of us to meet, discuss the successor to Menon. And I could, for the first time, see him function in such a situation. And it was quite, I learned a lot, say. He conducted a whole meeting with great sophistication. It was very disparate with various views, and he moderated it excellently. And that was the, and later on I had more opportunities of interacting with him in a quasi-professional way. I became a member of the Indian Academic Council when he was the president, and I could watch him conduct meetings, and the way he led the academy, it was an excellent lesson for me, but not much of practical value. I'm never ever going to be president of any of the academies. So, well, so that was the other time I had some kind of semi-professional well, on this occasion, I may also congratulate him on his son's achievement. Imran has been given the Infosys Award, and there is a Tamil, the famous Tiruppural has a saying. It says, essentially, it was, the greatest gift a son can give a father is for people to wonder, what parents did this man do to beget such a son? So I would also ask Obaid to convey my congratulations to Imran on his achievement. Well, as I said, my contacts were limited, not very much, but I enjoyed any time I spent with him, and I was particularly happy when occasionally he singled me out to join in the kind of West Canteen to talk mentally. So it was always a great pleasure. Well, let me end by thanking the organizers for asking me to reminisce here, and it has been a great privilege, a pleasure to have known Obaid and Asiya for 40 years and more, and I wish them many more pleasant, happy, and fruitful days. Thank you.
Thank you for that. I believe no reminiscence of TIFR is complete without Professor Sreekantan telling us some things. You may wish to sit there if you want to, or you could sit there if you want to.
Well, I came to know of Siddiqui's name even before he arrived in TIFR. And it happened in a very interesting way. One day, Dr. Bhabha came to the faculty meeting, and before anything else started, gentlemen, I have a surprise for you. We are going to have a molecular biology division in the institute. This, of course, there was a mixed reaction at that time, because perhaps as Daniel reflected his opinion, some people thought that why molecular biology in Tata Institute of Fundamental Research? It was thought that molecular biology is something that is not fundamental anyway. But then, you see, there was, Bhabha waited a little, and then he said, one young man by name Professor Siddiqui is coming soon, and he will initiate the program. But in the meantime, what had happened was the Bhabha Atomic Research Center had a biology division, and they were keen on having Professor Siddiqui there. So there was a kind of, Gopalangar was the head of the biology division at that time. He was trying to convince Dr. Bhabha that the molecular biology division should be BRC. So we were not sure what is going to happen. And I also remembered Bhabha, in one of his speeches earlier, had talked about Professor A.V. Hill, who was the president of the Royal Society at that time, who was also an advisor to Dr. Bhabha and also Pandit Nehru, he had told him when Bhabha had the idea of starting TIFR that biophysics should be part of TIFR. And Bhabha, in his usual style, had said, when the right man comes, I'll consider about starting that activity. And 15 years later, after Bhabha started TIFR, 15 years later, Obaid turned up and Molecular Biology Division started in TIFR. And we started hearing the name Drosophila quite often. Then several years later, when I happened to be the director, one day Professor Siddiqui came and said that he wants to have another institute devoted entirely for pure molecular biology without any applications. I found it a very wonderful idea, and I liked it. Then I took it to the council, and the council also welcomed it, supported it, because by then the molecular biology division had become quite well known within the country. But the opposition came from the Department of Atomic Energy. The DAE people said, why one more institute? And why out of, Professor Siddiqui was very keen that it should not be in Bombay, right from the beginning. So why outside Bombay? And Dr. Raghunathan was the chairman of the Atomic Energy Commission at that time. And he said, let him come to Indore, where we are starting the CAT. Or he can go to Pune, but not. So after a lot of persuasion, I think Professor Siddiqui also played a very important role in that. And fortunately for us, Professor Madan was there in the planning commission. So the constellation was right. Ramana in the chairman of the Atomic Energy Commission, and then Professor Madan being there at I think we succeeded. And it was decided that another biology division will be there in Bangalore. But of course, as Obaid knows, we had a lot of problems in getting a proper site, because GKVK was not agreeing to give the site, but then they agreed for 5 acres, 10 acres, and finally I think 20 acres or so could be managed. I'm happy to tell you that that is how NCBS also started. So the credit for the entire molecular biology activity of TIFR was entirely to Obaid Siddiqui, because right from the beginning, it was started as one man. Then, of course, Pavitra joined. So gradually the activity started. And I think today we can proudly say that both these divisions of TIFR are perhaps doing very well in this particular area. Of course, I'm not being a biologist. I'm not able to say all the achievements of the group, but I know that, in fact, Vijay last year got the Infosys Award. Mentioned about Imran Siddiqui. Last year, Vijay got the award, the same Infosys. So the tradition is maintained, and I'm sure there is going to be a great future for this activity in the country. And I wish Obaid and his wife and the family all the best in the coming years.
Thank you. Thank you. Can we ask Professor Divakaran?
This is becoming a nostalgia fest, so I'm a little bit wary about talking of the days when Ved was in Tata Institute, along with several people who are present here. The reason is, it is true that I'm a historian. I do more history now than art science, but I'm very careful. I pick my topics from several centuries back, and I pick people who are not in a position to talk back to me, especially at the first floor terrace of the canteen, over dawdling lunches. So, but on the other hand, this is an occasion for celebration. So I'll declare, I think nobody who knew him then knows him now will contradict my statement that Ved of today, here and now, is a pretty close approximation to the Ved of there and then. There being the Tata Institute in Bombay, and then being the 1960s and 1970s. See, big changes took place in those years. I mean, momentous changes took place, and two of the agents of change are present here, Ved and Govind. And before they came, TIFR had normal scientists. That is to say, people who understood most of what the other scientists did. But in those days, the universities taught botany, zoology, etc. But no subject called biology was taught in most universities. So when they began their activities in TIFR, they had to call upon us, the normals, to help in selecting, because they took students from all disciplines, because there was no particularly well-suited teaching program anywhere in the country from which they could pick well-prepared students. So they had to call upon us, the normals, to interview and select students. And these were absolutely marvelous occasions, because I've been there very many times, and some people here will remember seeing me at the other side of the table. And it was good for many reasons, because interviews lasted a long time, and we didn't mind it at all, because this being a biology laboratory, there were refrigerators in the lab. So that was a good thing. Obaid may or may not know these things, but this is true. The other thing is, we were very impressed. We actually discovered that biologists knew why a bird perched on a high-tension wire doesn't get electrocuted, for example. Or how to compute a complicated probability. Babu will remember. So this was a wonderful time, and we got to know very well one another. There were interviews, for example. Most people here know the story of the buffer solution, no? This was a student who Pavitra was pestering because he was a chemistry student. Lots of questions, and it turned out that he wasn't very well prepared for the interview. So finally, Pavitra said in desperation, he said, "Tell us what you know. We'll ask you questions on that." Then he stopped for a minute and he said, "I know about buffer solutions, sir." And Pavitra said, "Tell us about buffer solutions." And he said, "I know the second part of the answer, sir." So Pavitra persisted, he's a very persistent person. Then he said, "The second part of the answer is, such a solution is called a buffer solution." This actually happened. I was an eyewitness. I was there. I was present. So these, it breaks barriers, this kind of thing. And then, and also, at that time, TIFR was a wonderful, terrific blender of people and disciplines and so on and so forth. Not all people, not all disciplines, but on the whole, a very exciting place to be. That, of course, people have gathered by now. But the blending also took place outside. It's already been said that Obaid is a very fine tennis player. And that is true. He doesn't play anymore, as far as I know. He was a terrific player, and those who wish to understand how talents can be passed from generation to generation should study the Siddiqui family. I think some of them are. So in any case, we had a TIFR sports club. We also met them on the cricket field. And the molecular biology unit was a very fearsome team to face, because not necessarily because of their talent, but they had a team of cheerleaders on account of all the girl students. Is Shobhona here? So they beat us off the field. So these were truly wonderful times. And then, of course, Obaid and I played a lot of tennis, and Obaid is formidable. Whether you see him on the other side of the net or on your side of the net, he's formidable. So occasionally, once we got very brash. We said, TIFR Sports Club will participate in the local Bombay tennis tournaments. And so we played in the tournament, and we would win the first round, second round. Third round, we'll run into the club professionals, people who have been playing, who pretty much play tennis all the time. And we'll lose. And then we'll come back. And the next day, there'll be a headline, back page headline in the Times of India. TIFR scientists not up to the mark. or maybe even TIFR scientists bite the dust, things like that. So it's happened a couple of times, and but of course we took our revenge. We both of us went and joined the same club, played with them, and maybe even beat them once, I think. So this was life was very exciting, But it's very easy to lose oneself in nostalgia. I think what is wonderful is that I've been here just over a year, and what I find links it directly to the excitement and the spirit of adventure of the old TIFR is precisely the same feeling that nothing is impossible. We can do it. And I hope very much that it remains that way. It stays so. And I hope, personally, Obed, that you will be around for a long time. Make sure that it keeps that way. Thank you very much.
Thank you, Professor Divakaran. May I now invite Professor Balaram to say a few words.
I'm the only person to speak so far who's not been associated with TIFR. And I've also realized that it's very dangerous to be associated with Vijay and Krishna, because they sent me this program as an attachment on the email, which I never opened. And I never open things which Krishna sends me. And then my office had already marked it on my calendar that I have to go because I'm supposed to speak somewhere. So I'm here. I tell you, I was a bit pleased to hear Professor Sreekantan because he referred to Professor Siddiqui and not to Obed. And I was very uncomfortable because I've always known, at least in my mind, he's always been Professor Siddiqui. And I'll tell you why. When I came to the Indian Institute of Science in the early 1970s, shortly after I arrived, Krishna needed a supervisor, so he adopted me. He knew about TIFR because he's already shown you that he was at the BRC training school. He knew about TIFR. He knew about Professor Siddiqui, and he also knew about molecular biology. And he would keep on telling me, that's the place to be. And actually, when you sometimes think about Indian science, you might actually think about a tale of two Indian institutions, my own and TIFR. They have grown over a very long period of time, my own over a much longer period of time, and evolved in different ways, and they've had different cultures. So when Krishnan began to go to TIFR and eventually, I think, found himself a job in TIFR, I first saw TIFR sometime in 1977, just after he'd come back. And I remember going there, and I think many of you are much too young to know what the 1970s were like. In the 1970s, India wasn't doing too well. The science was still very difficult in most places. The emergency was on or had just been lifted, and there was no money to do research in places like the Institute. And TIFR was like heaven. It was the first time that I saw the West Canteen, that magnificent structure erected by Bhabha. And I realized that there was something in India which looked completely different from all the other Indian institutions that I'd seen. I also realized something else. Everybody called professors by their first name, at least in molecular biology. Not so sure in mathematics. Everyone called Professor Siddiqui Obed, and I was, of course, in the school which was headed by Professor G.N. Ramachandran, whose picture Krishnan flashed. And I don't think at any time any of us would have dared to call him by his first name. I believe he was called Ram by his friends, but I don't think any of us would have ever ventured to do so. I suspect there is still this cultural difference between institutions in India. But I also became aware of the fact that there was a move to create at that time what was called the National Biological Laboratory. And there were discussions which you would hear in the corridors. They were talked about at different places in India, and many new institutions were conceived. I think one of them was the NCBS. And I think I've heard about the NCBS from the time it was a gleam in Professor Siddiqui's eye, more or less. Professor Ramaseshan, who passed away some years ago, who was the director at the Institute, told me sometime in, I guess, 1982 or thereabouts, that Professor Siddiqui would like to create a new institution and he'd like to come to Bangalore. And it wasn't Indore by then. It wasn't any other place, but it was Bangalore. And he said, don't you think they should come to the Indian Institute of Science? I was young. And then I realized that the director of the Indian Institute of Science at that time probably had very strong opposition from many of his senior colleagues to any such move, like providing a few acres of land inside our campus for what was then the NCBS. But from that time, I have known NCBS and the manner in which it's grown, how it got its land here, thanks to the help of many people like Professor Sharat Chandra and others who worked very hard to get this land. And it's really spectacular to see NCBS the way it is today. And in some ways, I'm rather glad that it didn't come up on the campus of the Indian Institute of Science, because it has, I think, transported a little bit of the excitement that Divakaran spoke about, and a style which I think has been inherited from Bhabha's conceptions at TIFR many years ago. And I think it's been a real privilege to have seen all of this. I have been an observer from the sidelines of what happens in Indian biology for a very long time. And I've seen other institutions come up, the Center for Cell and Molecular Biology in Hyderabad, the National Institute of Immunology in Delhi, and many, many new biology institutions. But I think nothing's quite matched the style and performance of NCBS, and I think the credit here must go completely to Professor Siddiqui. But I will tell you about my visit in 1977 to TIFR. I entered and Krishnan told me, you have to attend a seminar. I went in and that seminar was Vijay. He was a PhD student then, and he was talking about Drosophila muscle development. And I don't think he's ever moved on. I didn't quite understand what he was talking about, but I've had the rare privilege actually of seeing both Professor Siddiqui and Professor Vijay Raghavan, who are really the architects of NCBS at different stages during its growth. And I'd like to wish Professor Siddiqui, Mr. Siddiqui and the family all the very best in the years to come. And I hope Professor Siddiqui has very, very more than the 5 years that he wants to do experiments. And I hope at least some of us will come back for Obey That 90. Thank you.
Thank you, Professor Balaram. May I now invite P.N. Bhavsar, who began his career in Obaid Siddiqui's lab, to come and share his memories.
Truly speaking, even at this moment, I have a conflicting feeling to speak or not to speak. Maybe I will elaborate a little more why I feel that way. I am one of the person who had very long association with him, almost right from 60s to till I retired in 2004. So I should express, but I am reluctant to speak. Those of my friends who know me, I am always hesitant and reluctant to speak, especially at this gathering, because somehow I have harbored the feeling I have very repulsive personality, or I at least isolate myself and don't interact with the people. So if there have been an interaction with my colleagues in general, or with Professor Siddiqui in particular, they are very limited. Secondly, I have a disappointingly poor memory. So even if I had interaction there are very few things to say. Okay, so with these things, I think I'll just do a compromise and I will say few things which I can recollect with some clarity. My first encounter with Professor Siddiqui was at the time of interview. I had passed my 12th class. I was desperately looking for a job. And in those days, probably getting job was not very difficult. And I got an interview call. Professor Siddiqui was was interviewing me, and he asked me a very simple question. Tell me whether air is heavy or vapor is heavy. And being not confident, or maybe out of fear of failure, I didn't want to give very clear answer. So I said it depends on density. So he said, all right, so tell me what is density. So I recited the definition of density. Then he was back again. Now tell me which is denser. So somehow, by that time I had made up my mind and gave the answer. And then there was a bumper. He said, now you gave the right answer. Now tell me, by sitting here, how did you work out that this particular thing is denser than the other? So there I could realize that here is a person who is trying to sense, I know the basic, and slowly and steadily guiding me to arrive at the answer, making me feel comfortable. The second incident which I can narrate to you is, after joining him, I had the initial training of doing microbial things, doing various experiments related to microbial genetics. And then he was away at, I think in 68, he went as a visiting professor to Cambridge MIT, and he had given me a project to isolate temperature sensitive mutants in E. coli. And somebody else was asked to supervise me. And somehow things were not working with me. Either I will not have bacteria growing on the plate, or I will have my plates flooded with contamination, or if things are working, I have no mutants. And the person who was supervising me used to make me feel very uncomfortable, almost not directly saying, but making me to feel that I am dum-dum, or I am clumsy, or I am plain lazy. Somehow 6 months passed, I had few mutants. I had isolated some mutants by that time, and Professor Siddiqui came back. I could feel the contrast, the way people interact with their assistants. So he came and he told me, I have come back, and let us talk about what have you done. So with a lot of bad feeling, I said, I don't have many mutants. So I said, I'm not asking you how many mutants you have. Let us sit down and let me know what you have done and how you have done it. And then I narrated him what all I have done. And then he probably dissected out everything, and he said, look, Bhavsar, it's not enough to learn or to acquire skills to do experiments. But in science, you have to learn to solve the problems. Now you have a problem. You are not getting mutants. You are using certain mutagenes. So you have to do growth curve, you have to do survival curve, and you have to see that you work out the optimum concentration of the mutagene, and then how to arrive at answer. So those are two things I clearly remember. I think I will stop here. I wish him on this occasion a very healthy and peaceful life ahead. And before I forget, I would also like to thank organizers for being kind enough to invite me on this occasion and giving me an opportunity to revive all those memories.
Thank you, Bhavsar. May I now request Dr. Babu to come and share his reminiscences. I think in some ways Babu is the physicist who started doing biology. So what we heard earlier on, that the physicists were had doubts about biology coming into this Institute of Fundamental Research.
Yeah,
Babu actually
I will
talks
talk
about something quite different.
First thing I would like to say is that I joined TAFR a year before Govind. Govind joined in 62, I was there in 61 as a student. 63, Maitra came. Obey had given us a colloquium on genetic code sometime, I think in 64 or so, I'm not quite sure. That was a very the talk was very, very interesting. All the physicists, most of us attended the colloquium, and it got us really excited. Of course, our ideas were rather simplistic. It was somewhat like Gamo's thinking or things like that. There will be mathematical solution to it. Obviously, there was not a mathematical solution. Biology is basically an experimental science. I only realized it much later. By the way, I should say that I am talking more as a prodigal son because I have tried too many things in my hand, never really succeeded. I will give examples of them and how it happened. One particular case I would say is that I had gone as a postdoc after working with Udgankar and getting my PhD. I went as a postdoc to Caltech and came back in 68. I came back at the same time when Anil Sadgowar also came, and he tried to tell me and excite me about the possibilities in biology. And I was feeling that I really can't do theoretical physics. I was no good at it. I could do some simple calculations, but I really was not cut out for that. So biology sounded very interesting, and I tried to read a bit, and then I went and talked to Obed. Obed readily said that I could work in his group. Not only did he say that I could work in his group, there were several things he did. One was there was a workshop on phages where Frank Stahl had come. In fact, the photograph was shown by you. That was a fantastic experience. I learned something about phages, except that I occasionally used to forget about adding the phase solution. Then on top of that, he said that I should go. He made arrangements for me to go and work with Sidney Brenner at Cambridge, MRC Cambridge. I was able to spend almost 3 years there. I would give you some picture of what it was like at MRC in 1970s, early 70s when Sidney was working there. Sidney was set up this C. elegans group. Sidney Brenner was furiously working on almost saturating the map of what he called uncoordinated, dumpy and various phenotypic mutations. John Charleston was diligently peering down the microscope with Navarsky optics, trying to figure out the complete cell lineage from one cell to 1,000 cell organism. There were about a dozen postdocs from US, all bright, very ambitious, trying to do various things in biology, in nematode development. I should mention that it was there was a dichotomy there. Sidney Brenner was also interested in looking at microscopic sections, electron microscopic sections, and putting together the whole nervous system. But there was a little bit of more focus on lineage, because it was clear that it will come through is much more interesting if it came through. I was there, quite dumb. I had studied only physics. I had hardly done any biology. I didn't realize how important it was to work on the lineage. I was reasonably okay with genetics. And of all things, I decided to work on the autofluorescence of the gut cells. I thought it's very simple to do. I can illuminate with some crude UV device. I can get mutants equivalent to eye color mutations of the software. You can use it as markers. Little did I realize that this is not exactly going to get me very far. I'm telling you this because the reason is that I've had several mentors in various areas of physics and biology, very famous. I realized much later that all the mentors must have suffered enormously. How to convince this fellow to do something constantly, steadily for some years to come so that he gets some results. He tries this, he decides on his own what he has to do. But of all the people, the worst sufferer was Obed, because he was my mentor for probably the longest period, over the first 20 years of my, I would say, youthful period when I was almost a student or just a faculty. Not only did I not pursue probably the interesting areas, I also decided Drosophila has much better genetics. C. elegans is crude, you can't get balancers, you can't do certain kinds of experiments. Let me do Drosophila genetics. So I came back to India, 73, and within about 2 or 3 years, I decided to switch to Drosophila genetics. I realized, I'm sure Obaid must have felt rather disappointed. Finally, he was able to get a new group going, doing C. elegans work, and this fellow decides that he wants to do Drosophila genetics. And what did I do in Drosophila genetics? I chose 2 completely esoteric areas. One was transfection. I don't think many Drosophila people would even heard of the word transfection. That has something to do with pairing-dependent complementation. You get complementation between 2 mutations if those parts of the chromosomes can not only complementation, some kind of interaction. This DNA-DNA interaction excited me very much. I had some ideas, theories, no real solid experiment. I didn't pursue DNA sequencing and so on. Simplest experiments, what one might call classical genetics, I pursued, and I got what I thought was some interesting results. But I stopped there. I didn't pursue it further. I didn't do the sequencing. I didn't try to see what kind of interaction was there. Didn't do the molecular biology of it. The other gene I got interested was wingless. I somehow felt that wingless must be very interesting because it turned out that it is basically a non-autonomous mutation. Almost all known homeotic mutations at the time were autonomous. So this was clearly telling something. This is non-autonomous thing. But I was able to show with some simple experiments that as a group of cells, it is not non-autonomous. Again, the results were very indirect. We did some EMS mutagenesis, showed that with EMS mutagenesis, we could get the autonomy part restored and so on. Didn't pursue it, left it at that. But it turned out WNT is probably one of the most important genes one can think of now. But coming to Obaid, I would say that he never showed his unhappiness. He was always ready, encouraging, will give me ideas how to go ahead, do the next experiment, and so on. He created an atmosphere in AFR which was very unusual, as Divakaran had pointed out. And Bhavsar had also pointed out. One of the important things he did was to put people at ease at an interview. He had various kinds of people joining biology, whether as students or as faculty. He was almost spot on in selecting the right kinds of students or right kind of faculty. I'll give 2 examples, because these 2 examples they will not feel embarrassed. One is P.K. Maitra, the other is Veroniki. Remarkable achievement. But as I told you, he was, even though he was more often right than not all cases turned out to be successful. I'm sure I and Krishnan must have given him a lot of. I think I'll stop at this point. Thank you.
Thank you, Babu. May I now invite Professor Arjun Singh.
Dear Professor Siddiqui, my colleagues in the past, friends, students, and everybody who is present in this room. Unlike many scientists, I believe in a supreme power who has created this universe, and in my view, perhaps many more universes. If you look around yourself or look inside an atom or subatomic particles, you see the beauty of this supreme power. If you look inside you or outside you, everywhere you find beautiful things. And I had the experience of looking down the electron microscope with the grace of Professor Siddiqui, and I could appreciate the beauty present in the structures of each organism. This supreme power is known by many names. we call in India as Brahma, Paramatma. In other parts of the world is known as Yehova, Ahura Mazda, God, Allah. In Hindu religion, there are deities who have booklets in their name with 1,000 names for that deity. So this supreme power brought a soul in this world around 80 years back. And this child was named as Obedul Haq Siddiqui, who grew with a very magnanimous personality, kind-hearted, discipline-loving, very noble professor, who founded the first Institute of Molecular Biology in Tata Institute of Fundamental Research. I joined the first batch of Atomic Energy Training School in 1957. And by my choice, I joined, after passing from the training school, I joined in the radiochemistry division of BARC. There I worked on the radiation chemistry and chemistry of trans-uranium heavy elements like plutonium, neptunium, uranium, thorium, etc. Out of these radioactive elements, plutonium was supposed to be very toxic, and right on the very first day, when I opened the consignment from Amersham lab and handled it inside the glove box, I was told that half a microgram of the plutonium only you can tolerate throughout the life. So you can imagine half a microgram, even a dust particle weighs more than that. So, and on top of it, plutonium is known to be a bone seeker. So once it gets into the body, there's no mechanism to throw it out. It goes to the bone and irradiates bone marrow, which could lead to cancer. After 7-8 years, there were 2 articles in Times of India about the cell biology and DNA, RNA by a person known John Osmundsen. I read those articles, and that inspired me to do a study of plutonium with bacteria so that I could infuse plutonium in the bacteria and leach it out by using certain chemicals or drugs, which I had in my mind. And in that connection, I started reading bacteriology books. Several of them I read, and I had sufficient theory knowledge about it. And I was feeling a little confident, but in the government organization, you cannot easily switch over to another subject. So I spoke to my head of the department, Dr. H.D. Sharma, Haridas Sharma. He was an exceptional person. He had worked with Glenn Seaborg, father of atomic bomb, and his PhD thesis, I could tell you, just contained 35 pages. The shortest thesis I have seen until now. I read that thesis 2-3 times, and I was very inspired by him. And then I went to him and told him, look, sir, I want to do this study with bacteria. So he was very encouraging. He said, yes, you can do it, but please do it in a biology department. So why don't you go and meet the director of biology in BRC? At that time, Dr. Gopal Iyengar was director of biology. I sought an interview with him. I went and met him there. He listened to me. And then first question he asked me, have you ever done biology experiments? I said, no, sir, I have never done biology experiments. He said, then you better remain in radiochemistry. And he did not agree to my idea of working there. I came and again told Dr. Sharma that, look, I had this interview and this was his response. Dr. Sharma said, you go to Tata Institute of Fundamental Research. A new professor named Professor Siddiqui has come. Go and meet him. He may be more lenient and receptive to your ideas. I came to TFR.
At that time, our labs were in the C block of DIFR. And Professor Siddiqui used to sit far away from the main lab. On the 6th floor, I think he was sitting in those days. I went and met him there. I explained why I want to do biology. He said, yes, you can come. Bacteria can be used for plutonium leaching. I felt very happy. He said, come after 3 or 4 weeks and you start working. After a month, I came there in the lab. The very first day he took me, showed me a bench. He sat down with burner, sponge and Lysol. He cleaned the table. He showed me how to handle bacteria and Aspergillus fungus in aseptic conditions in the lab. That was the first instruction from him. And he also said that you practice this so that you become proficient and you will have to work on the bacterial transformation of Bacillus subtilis. I picked up the techniques, started working with Bacillus subtilis. Fortunately, or with the grace of God Almighty, my experiments started working beautifully and I forgot about the problem of plutonium and leaching. You will be glad to know that I did my PhD under the guidance of Professor Siddiqui. I'm not sure whether I am the first student under his guidance from Bombay University or somebody else is there. That part I'm not sure, but probably I was the first student to get PhD from Bombay University under Professor Siddiqui's guidance. Things went on, and then in early 70s, the whole group decided that we should switch over to Drosophila, as Professor Babu had said. And 2 systems were approved, I believe. One was Drosophila and other was nematode. And Shahid Siddiqui was the person who carried the flame with nematode, and I believe he still is doing work with nematode in Japan or somewhere. No? Northwestern. So that part he didn't tell me. So as a whole group, we started with Drosophila, and Professor Siddiqui asked me to set up an electron and light microscopy lab for Drosophila neuroanatomy. He sent me on a training mission to same lab where Professor Babu had been there in Sidney Brenner's lab to learn serial section electron microscopy, a tedious and difficult job. I went there in Professor Sidney Bennett's lab, worked with John White, Dr. John Sulston, and learned serial section electron microscopy from Nickel Thompson. And as you know, Dr. Sidney Brenner and John Sulston both received Nobel Prize on nematode later on, after a few years. After setting up the lab, we had a group of colleagues whom I must acknowledge. The first colleague which joined our group was Mercy George, then Sunita Venkatesh, then Shubha Shanpa, Seema Shuralikar, Dr. Rajshekhar. These people have, and my wife Kusum, they all contributed in the progress of neuroanatomy at TIFR. We chose two systems. Okay, 2-3 minutes. We chose 2 systems, the gustatory system, where we had developed accurate maps of sensilla present on the legs, mouth parts, proboscis, and we completely worked out the ultrastructure of these and composition of these and produced maps for these which were useful for anatomy as well as for electrophysiology. Besides that, we developed cobalt injection and horseradish peroxidase injection techniques in our lab. And a powerful technique was developed in our lab where you could inject horseradish peroxidase and predict the function of the neuron by its staining properties. So you could identify a sugar sensing cell a salt sensing cell, a water sensing cell, and in very precise manner is projection by using horseradish peroxidase injections in the neuron. After that, besides that, we also worked on the olfactory system of Drosophila. We developed a very simple and powerful technique of identifying porous sensilla on the antenna, and we developed map of all three types of sensilla on the antenna. We determined the cobalt projections in the brain, as well as the whole DM-D1 whatever nomenclature you are using for describing the glomeruli, that was developed by us and Reinhard Stocker. Reinhard Stocker had come to our lab, and he had learned techniques from us and used this method with us for developing and marking the glomeruli in Drosophila. In this way, whatever progress we had made has been made by the blessings of Professor Siddiqui, which would not have been possible unless he was our mentor in that lab. We could work peacefully under his guidance, and I wish him and his wife and children a very peaceful, long, and happy life.
Thank you, Dr. Singh. May I now invite Professor Shobhona Sharma.
Am I the last speaker?
No, that's Shahid Jameel after you.
All right. Yes. So, I mean, I was just trying to gauge that. I seem to be one sole representative of the student era that came into TIFR. And I must say, Divakaran, I think you were there to interview me. And that interview, I recall, I was from chemistry. And I was prepared for the question, why do you want to do biology? So I had some answer ready, molecules, enzymes, RNA, so interesting. After all, life is chemistry and so on. So I remember Obed didn't buy all that. He kept trying to get me to utter the word regulation, which of course I had no knowledge of regulation. I knew enzymes were catalysts. So every time he would ask, but what is it that the enzyme does? And I would say catalysm, catalyst. It's a fantastic catalyst. You keep glucose and this thing, nothing happens, but you need. So anyway, that was the interview. I still got selected, so I suppose somewhere I must have answered a few things correctly. One of the best things about TIFR is, of course, how approachable people are in terms of asking questions, in terms of talking to them. And in general, I mean, just the day I joined, I had to go for some medical test at this OIC actually, which I wasn't familiar, since I wasn't familiar with Bombay. And there I was waiting at the bus stop at Bennett Villa, and I had no idea when the bus would come, and I was wondering what to do. When a car stopped and somebody gave me a lift. And later, only after I got into the car, I realized it was Putran, and the registrar of TIFR Institute. And just to think that somebody would notice that someone is standing at the bus stop, some young, a nobody from TIFR, obviously he realized that I was waiting at the bus stop and gave a lift. So that was my first encounter with how nice people were at TIFR in terms of, inclusivity. And of course, our unit, molecular biology unit, had this problem of being called very parochial and nepotism and all kinds of words, because we would always go to any matches, any TT match, or even in the canteen, one person would go and then all the biology students would join that table, drag chairs, and virtually empty out the rest of the canteen and club there. And that would often include faculty who would come in and join and discuss. And of course, we were all terrified of Obaid's question of, so what is happening? You know, this would, depending on whom he asked it, of course, it would matter. So I had joined, I was working on plant biochemistry, which was closer to chemistry, so I could, slowly transit into regulation and so on. But Obaid wanted Manmohan to do genetics. So he would periodically keep telling Manmohan, now you've done enough plant biochemistry, you must do genetics. And so he actually went for a sabbatical and did maize corn genetics, fantastic work. And then he came back and he said, now we will do genetics. And so I had just finished my PhD, so I was quite eager to start work on genetics. So there we had, and he had also learned molecular biology, by the way. He had done some southerns in those days, the first few years to do southerns. So then we had to do this cross between dwarf and giant corn plants. And we went to the terrace, we had a greenhouse there, planted those seeds. And the next day I come, the rats had eaten them. So first lesson in genetics. So we made these nets around it. Another week gone, the pigeons had eaten the plants. Somehow we managed to get just one set of dwarf and tall plants and do one crossing to do test some hypothesis, which anyway had not worked out. So that was the end of the genetics, plant genetics that I had done. I must say that we all benefited from discussion with Obaid, even though I haven't done any neurobiology or Drosophila genetics. And I am right now, I work with malarial parasite, but that also is linked to what Obaid did, because I wanted to work on nitrogen fixation, having come from chemistry, biochemistry, etc. And I was trying to get into Fred Ausubel's lab, who, of course, is well established and at that time was working on nitrogen fixation, who had said that, well, wait for a year and we'll see, and so on. So meanwhile, Ahmed Bukhari, who had come for a Mahabaleshwar course, was actually asking for candidates to do molecular biology in malarial parasite, because he was doing sabbatical in NYU Medical Center, where the first molecular biology of the parasite was done, and the rest is history. So once I joined there, then actually one didn't look back on nitrogen fixation. It was all parasite protozoan biology, which is what I came back with. And I think all of us, yes, one more thing I wanted to talk about Pratima, who was a physicist who was one year my senior, and of course we got along very well. But I remember she had also joined TIFR and had kept on asking Obaid, where is the physics in all this? She was a physics student, and she would keep, I mean, Maitra would tell her, you go and read genetics. And she would read some and then not like it at all. But I think some conversation with Obaid switched her, and eventually she did land up doing East genetics. But I don't know how exactly you answered the question of where is the physics in all this. But clearly he could convince many of us to come from other fields and join modern molecular biology and carry on with it. Thank you. And I wish Obaid and Asya and everyone a very, very long and fruitful life. And we look forward to years of, decades of celebrations.
Thank you, Shobhona. May I now call upon Professor Shahid Jameel.
Thank you. So I realize I'm the last speaker, and I'm the only one who sits between you and dinner. But I also noticed that everyone was having double serving of cake, so I'm safe here. Well, I have known of Obaid Saab for as long as I can recall. But I think our first meeting was in the summer of '78.
I've known of him growing up in Aligarh, from where his family lived and where my family lived, and we were distantly related as well. And somebody mentioned about him being a tennis player and all that. It's not just him, it's his whole family. And I remember his younger brother, Dr. Aslam Qadir, who was a faculty member at Aligarh, is a fantastic tennis player. And he was kind of my mentor on the tennis courts in Aligarh when I used to play for the university there. And his father, whom we called Qadir Nana, used to come to the court and watch us play and tell us a lot of wonderful stories of his times. So I've kind of grown up knowing the family in that way. So it was really the summer of '78 when I was accepted along with Utpal as a summer student at TIFR in the biology department. And Utpal, if you recall, Jagmohan Singh was also there that time, who was working with Professor Maitra. And I was assigned to work with Krishnan. And Krishnan mentioned all those leaking gels, and he was teaching me how to make the gels. So I spent a whole summer trying to figure out how to stop leaking of the gels. Finally made a few gels and ran a few gels on larvae. I don't think I could do anything more than that. But the whole environment at DBS was fantastic. The Bombay monsoon was terrible. I mean, I was coming from a place where you got rain, but you didn't get that much rain. So we were stuck at night in the department. We slept on the library table, and Zita used to cook us rice when the East and West canteen had both closed. I remember Satpal was there. He was the nocturnal Sardar who would only appear at night. So it was wonderful, and the whole ambience of the place was really very, very nice. I mean, I kind of knew I wanted to go into research, but that summer really gave shape to my plans. Well, I didn't do any genetics. I did biochemistry, and then from biochemistry I moved into viruses. And so many years later. And when I moved back to India towards the end of the 80s, in the 90s, I really had a lot of contact with him and with NCBS, and made many friends at NCBS. And Vijay has never forgiven me for that one summer in TIFR, and he keeps telling me that I keep coming back to NCBS because of the Stockholm syndrome. That one summer in TIFR. Nevertheless, Obed mentioned about the work that Abu came and did with me, and it's always been a dream to publish a paper with him, and I think the responsibility of that lies on Abu's broad shoulders now. If my dream is not fulfilled, he is responsible for it. So, and I hope, Abu, you will do your best to help me out here. Anyway, so it's really been fantastic, this whole celebration, and the way I've seen NCBS grow, it's really very, very nice. I've had a few heroes in my time, and I think you are one of them. What I admire most about you is your simplicity, your ability to communicate with younger people, the faith you put in younger people. And that is really evident from something that is not really part of the Indian academic culture. And that is to build an institute and then leave it to younger people to run. I think that's a very, very important thing that you have done in your professional career. So I wish you all the best. Many more years of health and happiness, because we want you to continue to advise us and inspire us, and also give Krishnan some more time to change and do other things, right? So thank you very much. It's been wonderful.
Well, thank you all very much. Dinner's outside.
Dinner's outside. Thank you very much.